Okay, along with the labor and maintenance, there are along with it is fixed charges and their working capital charges. So whatever kind of expenditure and expenses that has been made in order to build up that factory, and then all of that they're spending in their raw materials, consumables, labor, maintenance, in their employees, all of these charges, they all are something that is going to play an influence in how they develop the entire industrial process. And of course, different expenditures and operating outlay, these all are something that is absolutely dependent on how much money and how much cost a company has to spend. So if the fermentation process, extraction process, and developing the process is too expensive, then most probably the company is not going to develop. So whatever the company, businesses, factories, and all different processes that go into making different products is very much influenced by how the company is thinking of spending their expenditures and costs. Then fermentation products can be broadly divided in two kind of categories. It could be high volume, low value products, or it could be low volume, high value products. Now, what this first one means, high volume, low value product is something which is produced in bulk, and whatever product is produced in very high volumes and in very large bulk quantities is usually of low value. And one of the common example of this high volume, low value product is beverages, right? Beverages are not sold for thousands of rupees. These beverages, they actually cost very low. Juices, beverages, and malt drinks, and all of these, they are actually of very low cost, right? So this low value means the cost that goes in when you as a buyer buy some products. So it is actually of low value, meaning it is of low cost. So this is something which is produced in very mass quantities and very bulk quantities. So it is of high volume. Then the other category, which is low volume, high value products, is actually some pharmaceutical products and medicines which are prepared. And those medicines, of course, when you buy them on the shelf, on the countertops, they are actually very expensive. You spend a lot of money, but the quantity and the volume that you get in relation to the money that you're spending is actually very low. So it is low volume product which has a very high cost and a high value. So uh, very broadly speaking, fermentation products are naturally divided into either they are produced in very bulk amount, in very high volume, or they are produced in very low volume, and then their related costs and their related values, right? So the example written underneath is exactly the same that we have already discussed, that first category includes food and beverages, whereas the second category includes very fine, refined, purified chemicals or any pharmaceuticals that go into this category. Then food, beverages, food additives, and supplements. Most of these things are an example of the kind of fermentation products that are made. So a wide range of fermentation foods and beverages have been produced throughout the history. And why is this keep on reminding us of the historical record here is because we're talking about dairy products, and dairy products have actually been is being developed for ages now. So there is this activities of lactic acid bacteria in milk, which produces cheese, which produces yogurt, which produces sour cream, and also there's also a famous product known as kefir. Both of these are there to add flavor and texture and also to give the dairy product a long-term stability, right? So these dairy products, they differ, and they differ in terms of the texture that it provides, in terms of the flavor, how sour it becomes, and also the kind of preservatives it has and how stable it is over a long period of time. And then, if you very much know that yeast is very commonly used whenever we are talking about alcoholic beverages, whether it be wine, whether it be beer, and whatever it uses, it has these sugar sources. All these sugar sources are actually fermented by the yeast, and yeast actually utilizes the sugars, the carbohydrates, and then it converts it into ethanol. And by the way, ethanol is the only alcohol category chemically which is actually edible. All other alcoholic forms are actually poisonous, like methanol is poisonous, isopropyl, propyl. These all are alcohol classes, chemical classes of alcohol, but they are poisonous in nature, right? So it's only ethanol which is actually safe to consume. So basically, all these alcoholic beverages, they contain ethanol as a source of alcohol. And then most processors use strains of Saccharomyces cerevisiae and other strains of these yeasts are used as baker's yeast for bread dough production. So um, these three things are something we are actually we know of, right? So from ancient times we know dairy products, from ancient times we know ethanolic products, and from ancient times we also know that yeast strains are used to make bread dough and give rise to the bread and the kind of bread that we have. Then there are certain organic acids which can be derived from microbial actions. And when we say that they're derived from microbial actions, we actually mean having them fermented. So the very first human use was of acetic acid, and of course we know that when we're talking about acetic acid, we're actually talking about its use as vinegar, right? So it was produced as a result of oxidation of alcoholic beverages by acetic acid bacteria, and acetic acid bacteria uses the alcohol and converts it into acetic acid. So a further aerobic fermentation, it involves citric acid. We have already seen the example that production of citric acid is one of the milestone in different fermentation productions. And we have seen this yesterday as well, that citric acid is actually produced by filamentous fungus, which is Aspergillus niger, which has become a major industrial fermentation product for different numerous applications. And one of them, as I've told you, that citric acid is there as a preservative in almost any kind of canned and preserved food material that you purchase from the market. Then there are certain kinds of vitamin supplements, and that are also and can be produced by microorganisms. So you can use the same biochemical pathway which the microorganism uses to produce vitamins, to produce amino acids, and then these vitamins and any kind of supplements which are produced through fermentation can be used for human consumption if we are getting them very purified, or if not for human consumption, they can be used as an animal feed as well, right? So if you're maintaining farms, if you're maintaining animals and veterinary clinics and animals and cattle for all different purposes, then these supplements can definitely be used there, right? So when we're talking about supplements, there is something which one thing which is very important to discuss. That is a single cell protein, which is also referred to and abbreviated an acronym to SCP, the single cell proteins. So, what are these single cell proteins? These are microorganisms in which there is a very high level of protein production, right? So, microorganism and their strain has been modified so that they do an overproduction of a supplement, an overproduction of a particular kind of protein. Please, I would recommend that you mute your mics so that we have less of distortion. Okay, thank you. So there is this high level of protein production. So what it means that if the microorganism and its biochemical system is mainly dedicated for producing different kinds of nutrients, proteins, vitamins, amino acid, then that microorganism can be consumed as a source of supplement, right? So, if the microorganism is safe to consume, like probiotic strains, let's say we give probiotic strains to for human consumption, for treatment, for health maintenance. So, if we can make probiotic healthy bacteria to produce a high number of protein or high number of vitamin, then it means that that kind of probiotic bacteria can be used as a supplement. So it brings us to this idea of a single cell protein supplement, which is microorganism overly expressing the vitamin. And then it can be produced from a wide range of microorganisms cultivated on low cost carbon sources. Of course, this is one of the advantages of cultivating microorganism because they can consume any carbon source most of the time, right? So you can use a very low cost energy source, a nutrient source for the microorganism, and microorganism would grow on it and it will give you that protein. Or if you have caused it to produce any kind of vitamin, then it will be providing that vitamin. This very last point it says that the biomass and the protein extract from pure or mixed cultures of algae, yeast, fungi, and bacteria may be used as an ingredient or substitute for protein-rich foods and is suitable for human consumption or as animal fuels. Of course, this biomass can be used if the microorganism is a probiotic and it is safe to be consumed by animals or even by the humans, then the entire biomass can be used. And just in case you're going for a more refined version of the same supplement, then you can go for the cell lysate and the extract of that cellular extract, purify the protein, and then you can use it for consumption. And like said, you can use algae to produce, you can use yeast cells to produce, you can use fungi, you can also use bacteria to produce and give you the same kind of supplement, and this would also be characterized as a fermentation product. Then we also have these healthcare products. So in items providing human benefit, antibiotics is actually the center stage, right? So it kind of the most important compounds that we know are produced by industrial microorganisms. And basically, what are these antibiotics? Antibiotics are secondary metabolites. So, what are these secondary metabolites? That there is this main substrate which is converted if there is a chemical reaction going on. So, there is this main substrate which is converted into a product, but along with that product, they get several other byproducts in the process. So these secondary metabolites, these are just something produced, and these uh, secondary metabolites are something not very essential for the growth of microorganism, right? So these secondary metabolites and the pathways which are producing these secondary metabolites are actually not produced in the very active growing phase of the bacteria. So when these active growth phase is gone from is actually surpassed, when the biomass which is being cultivated within the media has reached its maximum, after that point onward the cells can dedicate itself to production of secondary metabolites. And once they do this, of course, we will get important antibiotics from most of the fungi, from bacteria, from actinomycetes, etc. Right? So, what do we know? We know that almost 4,000 antibiotics has been isolated, and about 50 are used regularly in antimicrobial therapies, right? So, this is actually a very huge amount which has been produced, a very huge amount of antibiotics, right? And the most commonly known and the most commonly prescribed ones are penicillins, cephalosporins, and tetracyclines. And the thing is, this quest of finding antibiotics, it has not kind of ended, right? We know that we're continuously in need of new kind of antibiotics. So this quest, the search and research of antibiotics is very much concentrated on getting new kind of microbes and testing whether these microbes are producing any kind of chemicals which are antimicrobial or not, and then we're trying to identify different antibiotics. So there is a lot of research in this area which is going on. So other important pharmaceutical products which are derived from microbial fermentation or biotransformation are alkaloids, steroids, and vaccines. Okay, are you with me?
Yes, ma'am.
Okay. I thought I lost the connection for a while.
Yes, ma'am.
Okay, so there are a lot of pharmaceutical products which can be produced, which includes different alkaloids. Alkaloids have different kind of actions. They are nitrogenous organic compounds, and most of them are from plant origins. Like morphine is a very common, example of an alkaloid. Then there can be steroids for medicinal purposes and vaccines which are produced for medicinal purposes. So one of the examples of these biotransformation could be of morphine, which is converted into normorphine for their, opioid activity and less habituation and addiction. Like we know that morphine is used as a painkiller as well, but we also know that it is notorious, and it's very notorious drug because people actually get addicted to this drug, to morphine. And then there, you must have also heard of normorphine, and you use normorphine because it's kind of a modification of the same drug, and you do this modification through biotransformation, through a chemical change brought by the bacteria or the yeast cell, right? So we're using the biochemical pathway these microorganisms have to change the structure slightly and to modify the product that we already have. So they convert morphine into normorphine. And why? Because normorphine is much better. It's actually better than morphine because it leads a person with less habituation, right? Habituation and addiction is actually the main factor of morphine. So normorphine is also addictive, but it is actually less addictive than morphine itself. So there's different uses. So you're modifying different drugs. Same way, same way, steroids can be modified. They can be biotransformed. So as you know that all the steroids have certain main um, carbon skeleton, and if you modify that carbon skeleton with different in different classes, then you get different steroids in hand. So this parent steroid is biotransformed to give you very much of derived other forms of steroids which can be used. So more recently, a therapeutic recombinant human protein such as insulin and human growth hormone has been produced by a range of microorganisms, right? So we do know that previously insulin was something which was produced with in cattle and in goats and buffaloes. So we were using that animal system to produce insulin and then give it to diabetic patients. But then there was a revolution that took place, and that revolution was that insulin was recombinantly produced within the microorganisms, right? So now we have a recombinant insulin produced in bacteria, very economical and very much functional, and also very pure form of insulin that can be provided. Similarly, there are a lot of growth hormones which can be produced by a range of microorganisms, right? So we need hormones and we need production of hormones for different kind of endocrinological disorders. So if we can produce hormones and we can produce human hormones, we can cut those genes, we can recombinantly modify the microorganism and make the microorganism express that human hormone. So we can have these hormones as well. So there is this field is actually very much expanding, like we discussed yesterday, that DNA technology has actually revolutionized the way we used to look into biotechnology, the way we used to look into industrial product formation and all other things, right? So and this is actually a promising field, and this promising field is likely to make all other sort of contribution in the process, right? Then one more, another important thing is these microbial enzymes production, right? Like we discussed, that there can be a range of products that you can produce. We've seen that pharmaceutically you can produce different kind of products, um, and likewise, enzymes are very much important in all sorts of industries, right? Enzymes are important in food, enzymes are important in any biotransformation process, enzymes are important for these pharmaceutical industries. So it's actually very much important. So how can we extract enzymes and how can we produce enzymes in bulk? All of this is also contributed by microorganisms. We're going to make microorganisms like they express different proteins, like they express different other products. They are also expressing enzymes. We know if they are running a biochemical pathway, they are producing and utilizing enzymes. So if we want to make enzymes, we can use the same cell base to produce for ourselves all sorts of enzymes, right? So enzymes can also be produced through the process of fermentations. So particularly there is an extracellular hydrolytic enzyme have numerous roles in the process in the production of wide range of food and non-food industry as well, right? So most of them, what they are extracellular hydrolytic enzyme, and extracellular meaning they are expressed inside the cell, but then they are excreted out, right? So so the cell is produced, but the enzymes they are extracted and released into the medium. So it's an extracellular enzyme. So one of the examples of microbial enzymes is proteases. So, for instance, are extensively used as additives to washing powders and for the removal of protein from different other food and beverage products like beer, and also as microbial rennet for the production of cheese. As you know, that rennet is a kind of enzyme, and this rennet is actually extracted from different animal sources, and most of them also from the pig source as well. So, what are these proteases? Proteases are hydrolytic enzymes which break down proteins. So, this kind of property is very important in washing powders because if you have, these enzymes has power to cut down and remove any other, food related stain that you have on clothes and every other stuff which can be washed away through washing powder. So you can use hydrolytic enzyme to make the washing powder and surfactant more effective. And you can use these proteases in the removal of protein. And why in the removal of protein? Because they are hydrolytic against the proteins. So you can use that for purifying the product. Meaning, if beer is one of the products and it has a protein as an impurity, then you can use proteases to cut down and break down the protein. So it makes the purification and extraction process a lot easier. And of course, proteases are enzymes, and rennet is an enzyme which is used to make cheese, very famously cheese. So, so you can use these enzymes, which are essentially proteases, for different uses, right? So there's this washing powders, there's this purification of different products that are made, and also for the production of cheese as well. Okay, there are several carbohydrates, hydrates which are also produced. Like in previous slide, we were discussing about the proteins, proteases. So there can be enzymes which are particularly there to digest different sugars, right? Carbohydrates are sugars. So carbohydrates, they are employed in the production of diversity of sugar syrups from starch, right? So as you know that the starch is actually a polymer, right? And different sugar syrups is actually, disaccharide form of the entire carbohydrate system. So if you starting with a polymer and you are using a carbohydrate and you are cutting down the polymer, you end up with minor sugars, right? Like disaccharide sugars that we know of, that like sucrose, like fructose. So all of these can be produced from a starting material, which is starch. And do keep in mind that starch is something very easy to obtain, and starch is something very cost effective and cheap as well. So if you're using starch as a beginning source, and then you're using these enzymes to cut down that starch, and you can produce sugar like fructose and glucose and sucrose, then you like industrially, it is something very cost effective. So what happens that high fructose corn syrup is actually produced when you hydrolyze cornstarch, right? And you hydrolyze cornstarch to glucose using amylase. And keep in mind this amylase is an example of carbohydrates, right? So I assume that it is obvious, but what I'm gonna do is use a same color, so it just becomes more obvious. So we're talking about amylase, which is a carbohydrate, right? um, and what do you get? Resulting is glucose is then isomerized to a sweeter molecule like fructose, right? So what do you have? You have an amylase and you have an amyloglucosidase. Both of them, they are carbohydrates. So you use these two enzymes to digest the starch, and in return, what do you get? You get your sweeter form of sugar, which is fructose, right? So, so what do you have in your hand? That you have a starch solution which is cheaper to obtain, and then you have a much sweeter syrup which is used in making different kind of juices and different kind of products. And what do you have? You have a industrially used product which is commonly used, right? Now, the use of corn syrup and is basically an example of bulk enzyme consumption, right? Then there are smaller quantities of purified enzymes that can be used, and they are used for very specialized purposes, and most of them are for producing any kind of pharmaceutical product, because for the purpose of making an fructose syrup is actually the purpose is not to get a very refined form of product. But if you're making any kind of chemical, and if you're making any kind of pharmaceutical product, then use of very fine enzymes can be something very important, right? Now we have a very just few minutes left, and I'm thinking that we're going to continue at 7 p.m. Actually, the timing that I've given you on the link is 7:10. So I'm going to close this session now. If you have any question, please go ahead and ask. And if you do not have any question, then I'm going to end the session. I'm going to start at 7:10, inshallah. So if you have any questions, please go ahead, and then we're going to continue from this slide onwards in the next session. Is this okay? I guess it is okay. Anyone? Anyone who's there?
Yes, ma'am.
Okay, okay. Then I'm ending now. I'm ending now, and inshallah, we're going to meet in this next session.
Okay.